Hello everyone, I am recording this video to show you how to collect the information you need for your gene sequence. So previously you were to choose your gene of interest. You had to make sure that it was a protein coding gene, and you had to make sure it was from one of the four organisms that we listed. So the next step is to look into the gene sequence itself, find out a little bit more about where is it located, what chromosome is it on, and so I'm going to show you here how to gather that information. So I'm back here on the gene records page. I'm back to the GRM6 gene page in particular. And what you're going to be looking for on this page is information about um, uh, alternate names for the genes. So there are sometimes alternate names. They'll say also known as. So here, for example, this gene is also known by these other names. Um, and so I'll get you to write that information down. And then scroll down into the next section here, which is the genomic context section. And here you're going to find the location of your gene. So in this case, this gene is located on chromosome 5. It's on the Q arm in the section 35.3. It will also show you how many exons there are. We're going to record that in the next part of the project, but this is the information found here. And then you'll notice here that there's a list of assemblies. You don't need to worry about this section too much. What might be useful are the values here, these numbers. So these numbers here represent the nucleotide positions on the chromosome itself. So what they do to keep track of all of the nucleotides in the chromosome is they number them. And so this is the range within chromosome 5 that the GRM6 gene is located. And you'll notice that these numbers here should match or closely match the numbers that are listed here. But I would always reference the genome data viewer for the exact numbers if there are any discrepancies between these numbers, which I can see is a little bit here. I suspect this screenshot picture is from one of the previous assemblies. So what else you're looking at here are some of the other genes that are located nearby the GRM6 gene. So it looks like there's one here, ZNF879. There's our gene there, the GRM6. And you'll also notice that there's some arrows as well. So the arrows indicate the direction of transcription. So the direction in which the sequence is read. And so some of them get read from left to right and some get read from right to left, as is the case for the GRM6 gene. So the next place that I would recommend you go is into this genome data viewer. And I've opened that up here in another tab. And it brings you to a page like this, which gives you lots of great information and some really nice visuals as well. So what you're looking at right here, this is the chromosome with all this banding. And you can see here, this is the P arm. And then we go into the Q arm here, and all the numbers are all listed and labeled. And for your gene, what you're going to be looking for is a small purple line, as we see right here. This purple line shows the location of your gene. So this one, GRM6, is located right here in section Q35.3 of the chromosome. So then in this graphic view section right here, which is quite similar to the one on the previous page as well, you'll see there is the sequence listed here. And what you can do is you can hold your mouse. I believe it has to be collapsed for this to work. So if you hold your mouse over the line, it has to be collapsed. So you'll notice I expanded it. You can click on it. It expands into different sequence forms here, but we're not interested in expanding it yet. We'll do that next week. So if you hold your mouse over the green line without expanding or clicking on it, you'll notice that a whole bunch of information comes up. And this essentially summarizes a lot of the details about this particular gene. And throughout the next few parts of the project, we are going to look at more of these. But for this week, what I want you to look for is something that says the span on NC. So it's about 6 or 7 lines down in this box. And for this particular gene, the span on NC, remembering that the accession numbers that start with NC means you're talking about the chromosome sequence, that span is almost 17,000 nucleotides long. And so that's the nucleotides on the chromosome. So that's what I want you to record for this week. And then you're also going to record the chromosome position numbers that were listed on the previous page. So you can see they're listed right here as well. So it's showing what position it starts at in the little bubble, and you can scan to the other end and you can see it as well. I have to scroll my screen over to do that. So what else does this page show you? Well, it'll show you different exons. So as I hold over a bubble here, it highlights the different exons that are in this gene, and you can see it labeled just above my mouse there as well. So this gene has 11 exons as was labeled, and you'll notice in this case the numbers are going from 11 down to 1 in this direction, because don't forget that for this particular gene, there's those arrows. The direction of transcription starts on this end and finishes on this end. So it's read from right to left in this case. So that captures the information that you need to gather for part 2. If you have any questions, let us know.